The last group of viruses we will discuss are viruses which use reverse transcriptase, and there are two families, retroviruses and hepatnaviruses. Retroviruses contain single-stranded RNA as a genome, and upon entering to target cell, this RNA is retrotranscribed into DNA. DNA is targeted into nucleus and integrated into the genome. Hepatnaviruses are different. They use reverse transcriptase differently. Their genome is circular, partially double-stranded DNA. It has certain gap. So when they infect cells, the DNA enters nucleus where the second strand is completed and DNA persists in nucleus as an episome. It does not integrate. So where do they use reverse transcriptase? When RNA is transcribed, this RNA, when it enters cytoplasm, is afterwards retrotranscribed into DNA. So both use reverse transcriptase, however, at a different time and with different purpose. It was suggested that retroviruses are actually relics which witness transition of RNA world into DNA world, and still have in our genome so-called endoretroviruses. We will discuss that later. Now, let's start with retroviruses. The retroviruses variant is spherical envelope. An envelope is derived from cell membrane, and within envelopes they have the protein, surface proteins which bind to target cell receptors. They also have transmembrane protein which is involved in the fusion with target cells. A capsid is usually icosahedral or conical shape. They also have matrix. They package reverse transcriptase, and also they package integrase and protease in the variants. And the genome is single-stranded RNA. However, retroviruses always package two RNAs, two single-stranded RNAs, and also cellular tRNA, which serve as a primers during retrotranscription. Now, these proteins are derived from GAG-POL and ENV genes. So you can see it here. So the genome is linear, single-stranded RNA. Even though it is positive sense, it cannot dock on ribosomes. It needs to be retrotranscribed and DNA is targeted to nucleus. If you look for genome on each 5 and 3 primes, they have these repeats, and on 5 prime there are unique sequences and unique sequences on 3 prime. These three major group of genes are GAG-POL and ENV. However, before we go to their function, we have to also point that they have cap on 5 prime and poly-A tail on 3 prime. Now, these three major genes group encode GAG nucleocapsid, matrix, and capsid. POL encodes for reverse transcriptase, protease, and integrase, and envelope for transmembrane and surface protein. All these gene synthesize polyproteins, or respectively encode polyproteins. And some of the groups, such as lentiviruses and HTLV virus, they also have some additional regulatory genes. Now, let me come back to this. Most of these genes are non-spliced. GAG and POL are non-spliced. ENV is spliced. And remember, it's very important for maturation that the proteins which they encode are synthesized as a polyprotein, which are cleaved only later on. Now, there are 7 genera of retroviruses. There is alpha oncoviruses, beta, gamma, delta, epsilon, they are all oncoviruses. There are lentiviruses and spumaviruses. Oncoviruses, they carries a gene which can trigger cancer in the target organisms, such as there is a Rao sarcoma virus, mouse mammary tumor virus, murine leukemia virus, and so on. Lentiviruses, lenti means slow. Those are slowly progressing viruses, which actually eventually can cause very severe wasting disease, such as HIV, SIV, EIV, FIV, many other viruses. Spumaviruses cause persistent infection. However, there is no pathogenicity. Pathology was not detected. And for example, simian foamy virus. And here are all these viruses. So for humans, HIV, lentiviruses, viruses are highly species specific, so HIV only infect human, SIV only infect monkeys, FIV only infect cats, and so on. Another virus which infect humans is HTLV1 and HTLV2. They are both delta retroviruses. Now, disease, we know, we will focus in more detail this week onto HIV, which can lead to AIDS, acquired immune deficiency syndrome if untreated, HTLV1, HTLV2, they can trigger leukemia. SIV, FIV, and PIV, so simian, feline, and bovine immunodeficiency viruses can cause similar disease like AIDS in animals. Remember, all these are lentiviruses are species specific, so they do not cross barrier between species. And oncoviruses cause variety of cancerous disease in many different animals, starting from mice through cats, sheep, birds, and so on. Now, what is very distincting about the retroviruses, they have very unique replication cycle because their genome is RNA. So when they enter target cells, this RNA is retrotranscribed into DNA, double-stranded DNA, and this double-stranded DNA is transported into nucleus and integrated into the genome. Also, several of these oncoretroviruses, such as Rao sarcoma virus, trigger tumors because they acquire cellular proto-oncogene. They mutate it and basically convert it into the oncogene. So whenever they infect target cells, they can trigger oncogenesis.